As host, it's my job to introduce our speaker, just like Reagan did a wonderful job of last week with Dr. Sale. And so it's my pleasure to introduce our first speaker, Araya, and she comes to us from Brookwood High School over in Lawrenceville. I'm also a Gwinnetian, Collins Hill, so go Eagles, go Broncos, right?
It's actually Berkmar.
Oh, I'm sorry, Berkmar. Also Gwinnett. It says Brookwood.
That's mine.
Oh, Michaela, we got two Gwinnetians tonight. I saw that today. Oh, there we go. Okay. Berkmar, right. So another thing about being a host is be prepared. Anyways, but it's interesting to point out that I try to kind of mix up the information that I convey. And so she's got some stuff about here that she's traveled rather extensively, originally from Eritrea, London, Sweden, Canada, Denmark, France. But I think the other opportunity here is for students to talk about and for me to introduce their interests. And so she's been doing research over at Mercer with Dr. D'Souza, right? Yeah. And so this is a great opportunity to talk about what you're interested in. Michaela will be talking about some aspects of marine biology, which is interesting to her. And this is the work that she's been doing over at Mercer. And so I think that's it in terms of introductions here. And so her title is The Development of a Cross-Protective VLP-Based Influenza Vaccine. I give you our first speaker of the semester. Take it away, Saran.
So hey guys, again, my name is Araya, and I'm a biomedical research intern at Mercer University College of Pharmacy. And I work in a vaccine development nanotechnology lab where we conduct preclinical pharmaceutical studies to basically develop vaccines for different infectious diseases like influenza, gonorrhea, and even some cancers like ovarian, prostate, and breast cancer as well. So over the summer and throughout the semester as well, I'm working with a graduate student, Keegan Brace Gomez, where we're developing a cross-protective and universal virus-like particle-based vaccine for influenza, and I'll be sharing that data with you all today. So this is just an overview of my long talk today. So first I'm going to give you guys some background information on viruses in general, and also general information on vaccines and how they work. And then I'm going to introduce our research study that we conducted and talk about the two approaches we took. So we had an in vitro approach outside of an organism, and in vivo approach in an organism where we're using mice as our modeled organism. And then I'll conclude with our results and data and give you guys some key pointers to take with you guys today. Okay, so a virus is an infectious agent that replicates in living cells and can be very infectious. And there are many different types of viruses, including bacteriophages that infect bacteria, and then there are also viruses like influenza that affect eukaryotic cells. So there are three main components that are in common within all different types of viruses. The first is a nucleic acid genome. So it consists of either DNA or RNA, and that's the genetic information of that virus. And then there's a protein capsid that basically surrounds the genome to protect it. And then lastly, there's a protein coat, as we see here on the influenza virus, that is basically different proteins like glycoproteins and matrix proteins, which I'll mention again later on, and that assists with the binding of the virus to its host cell and help elicit a response in the immune system. So the flu is caused by influenza viruses that infect the nose, throat, and respiratory tract, and cause a viral infection that's very common and can be very deadly. In fact, last flu season, 2018 to 2019, over 62,000 people died just based off this influenza flu. So it is actually a lot more deadly than people may anticipate it to be. And I actually want to talk about the process of how viruses actually work and leading into actually killing organisms or people. So first, the virus binds to its host cell. And the first thing it does is it injects this DNA or RNA into the host cell, into that nucleus. And then that DNA is then replicated to create other viral particles. And those extra viruses basically replicate and continue to damage that specific cell. And so this is an ongoing process in our bodies that, and if we don't take proper precautions to alleviate this by vaccines, then they basically damage more and more cells in our bodies, leading to potential death. Now I want to briefly touch up on how vaccines work. So when you do get vaccinated, when you go to the doctor, usually by an injection, you get a weakened version of a disease or a virus that's injected into your body. And the reason why they use a weakened version is so, of course, that you won't get the actual disease that you're trying to prevent. And so your body can generate a memory response to this specific virus so that when you are exposed to it again, your body will have, or your immune system will have a stronger response against it. So again, you get injected a weakened version of the virus, and then your immune system assigns specific antibodies to this pathogen that's coming into your immune system. And these antibodies, which are Y-shaped proteins, also known as immunoglobulin or IgG, they are assigned to fight against this specific pathogen so that when you're exposed to the actual virus later on in your life, then it could properly fight against these pathogens and have a stronger response against it. Okay, so viruses in general are constantly mutating and constantly changing due to evolutionary pressures and viral genetic drifts. And because of this, researchers are always having to create different vaccines for the newly mutated versions of a specific virus so that it could be more effective for the coming years. So usually they update flu viruses every year or every 2 years to improve the effectiveness. And this could be kind of problematic because if we're always changing the vaccines that we're using for the influenza virus, that means the effectiveness of this is also changing, meaning some years it may not be as productive or as effective as other years, and some years it may be effective. So in our lab specifically, our goal was to develop a cross-protective vaccine that's going to fight against all the different strains of influenza, so we won't have to modify it every year. So cross-protectivity, again, basically means that not only are we trying to fight against the native strain of influenza, but we're trying to develop a vaccine that's going to fight against all the mutated forms and all the different strains of influenza that are created due to evolutionary pressures and viral genetic drifts. So this is a cross-section of the influenza virus here, and it does consist of a lipid bilayer membrane, and it also consists of surface proteins, which I mentioned earlier, on the surface of the virus that assists with the eliciting a response in the immune system. So the two major glycoproteins present are hemagglutinin, also known as NHA, and what this does is assist with the binding of the virus to its host cell. And the second major glycoprotein is neuraminidase, which is right here, also known as NA, and that facilitates the transportation of the virus throughout your respiratory tract. And along with this, there are two matrix proteins, M1, shown by these purple circles here, and then M2. So based off previous research, it is known that the glycoproteins HA and NA are rapidly changing and constantly changing actually across different mutations of influenza. However, the matrix protein M2 is known to be highly conserved, meaning it's not changing as much with different strains of influenza. So in our lab, we wanted to really focus on the M2 protein because this is going to be the common factor within all the mutated forms of influenza. And since we're trying to develop a vaccine that's going to be fighting against the newly mutated forms, we want to focus on this protein. So what we decided to hypothesize is that the M2 protein would create the most suitable cross-protective influenza vaccine. And again, the reason why we hypothesize this is because, one, M2 proteins are simple in its form, and it's going to stay consistent when it's mutated and exposed to evolutionary pressure or viral genetic drifts. But of course, M2 proteins alone are not going to elicit a response in the immune system. So we're going to look into virus-like particles. And what a virus-like particle is basically a particle consistent of multiple repeats of proteins and that resemble a specific virus, a native virus. So in our case, we purchased a virus-like particle which was made by a lab at Georgia State University. And this virus-like particle had multiple repeats of the M2 protein that's highly conserved that we're focusing on, and it mimicked this whole structure of the influenza virus. So even though we did purchase this, I still want to go into detail of how it's produced, so you guys have a better idea of what it's composed of. So this is our M2 protein, which is highly conserved and is represented in this portion of the influenza virus. And that cannot elicit a response in the immune system alone. So they're going to make multiple repeats of this M2 protein, and it's going to self-assemble by protein folding to create this structure here, which is called an M2 virus-like particle. And again, this virus-like particle is just composed of M2 protein, but it's going to resemble our influenza virus. Okay, so now I'm going to give you guys an overview of the study before I go into detail of each step. So again, this is our M2 virus-like particle, which was purchased from another lab at Georgia State University. And then we're going to conduct a chemical reaction with biodegradable matrix and insert it into a spray drying machine, which is a technique used for microparticle formulation. And I won't focus too much on this process since I want to focus on the actual study. But basically what the spray drying machine is formulate our M2 microparticles for our vaccine, which is shown here. And then we're going to do an in vitro approach to test for cytotoxicity of our vaccine and also for immunogenicity. And then we're going to do an in vivo approach and inject our vaccine into our modeled organism, which is mice. Okay, so for our in vitro approach, the first thing we did was test for cytotoxicity. And the way we did that was using an assay called an MTT assay. And this is basically a technique, a colorimetric technique used to detect for cell viability by detecting cell metabolic activity. So what we're trying to test is to see if our vaccine is going to be if it's going to be safe to inject into an organism. So we have to make sure it's not toxic to cells in the body before we inject it into the organism. And like I said, this is a colorimetric test. So the reagents we use actually conduct a purple color seen here on this plate. So the more purple color we see, the more cells that we have viable. Along with this, we do an immunogenicity test using nitrite assay. So the reason why we do a nitrite assay is because based off previous research, it is known that vaccines that are highly immunogenic in the body, meaning it's going to generate an immune response, they actually release high levels of nitrite oxide when they interact with viruses. So we knew that if our vaccine is going to be developing or generating high levels of nitrite oxide, we can correlate that to it having high immunogenicity, meaning it will create an immune response in the body. So what we first do is plate our cells, and we're just using regular dendrite cells in this case. And we're going to add our formulations to the M2 microparticle formulation that we generated for the vaccine. And then we're going to add the proper reagents and put them on a plate reader where it's going to give us that data through a computer software. So this is our results for the MTT assay. The y-axis represents cell viability, and the x-axis represents microparticle formulation. And so our negative control was DMSO, and DMSO is just stands for dimethyl sulfur oxide, and this is our solvent we use to dilute our vaccine. And cells only is going to be our positive control, and that's just the cells that are untreated, and we expect that to have high levels of cell viability since they're not treated at all. And then everything else is basically our vaccine with different concentrations since we dilute it with the DMSO. And we conducted the MTT assay the first 24 days, and then again after 4 days. And this is the results we came up with. So because our p-value was below 0.001, we decided to conclude that and since we have high levels of cell viability after our MTT assay, we concluded that our vaccine is non-toxic to cells and it's safe to inject or deliver in organisms. And before we actually decided to put it in organisms, we had to test if it's going to be generating an immune response. So it is safe to put into an organism, but we still need to see if there's going to be a response happening in the immune system specifically, since that's the whole purpose of our vaccine. And this is our data for the nitrite release assay. And again, vaccines are known to have high levels of nitrite release from the interaction between the vaccine and the viruses. So our y-axis represents nitrite and micromolarity, and our x-axis represents the different groups. Our negative control is just going to be cells only, where they're not treated and they're expected to have low levels of nitrite oxide released. And our positive control is the measles vaccine, which is known to release high levels of nitrite oxide, and it's known to also be highly immunogenic, so it's going to generate an immune response. And everything else is basically different components of our vaccine, and this is our main focus here, which is the M2 microparticle vaccine that we created in our lab. So what we predicted to see is that our M2 microparticle vaccines will have similar levels of nitrite being released than our measles vaccine, which is the positive control, and which is what we've seen based on our data. So as we see, our positive control has similar levels of nitrite being released when it's interacting with the virus, which is what we've seen. So this allowed us to conclude that our vaccine is non-toxic to cells, and is also going to generate an immune response in the body. So now we're ready to go to the next step and injecting them into the mice. So in our in vivo approach, the method we used to deliver our drug was a laser ablation method, which is a form of delivery through the skin transdermally. So we use this laser right here, and how it works is it produces small micropores with specific sizes and depths to the skin of the mice. And then that allows us to add our bioagents or our vaccine, the M2 microparticle vaccine that we created, which is shown by this figure here. And this is the different groups of mice we had. So we have our naive, which is our negative control with no treatment. And then we have our positive control, which consists of the mice that were injected with an inactivated form of influenza vaccine. And then we had mice that were just treated with just a laser treatment, so laser ablation method with no vaccine. And then we have a blank where they received unloaded microparticles that didn't consist of our vaccine. And then this is our main focus, which is the M2 microparticle vaccine that was generated in our lab. So we expect the positive control to have similar values to our vaccine. Okay, so this is a study schedule for the in vivo approach. Overall, it took 12 weeks to conduct, and the linear axis represents the week number. So we initially gave them a prime dose of our vaccine, and then in weeks 3 and 6, we gave them a booster, so we exposed them to the vaccine again. And then weeks 4, 7, and 10, we actually drew blood from the mice and isolated serum to test for immunoglobulin, which again is the antibody that neutralizes pathogens. And I'll talk more about that method in the later slides. And then finally, in week 12, we challenged the mice. And here we just exposed them with a live strain of influenza to finally test if our vaccine is effective. Okay, so when we isolated the blood from the mice, we isolated a serum, and this serum consists of antibodies that fights against pathogens coming into the immune system. And the way we quantify this is using an assay called enzyme-linked immunoabsorbent assay, also known as ELISA. And the way this works is it detects the amount of antibodies you have present in your substrate. In this case is our serum that was isolated from the mice blood. So we initially had a plate like this, similar to this, and in each plate we added our M2 microparticle vaccine, which was our target protein. And then we added the serum, and the serum we predict has the antibodies present that was already assigned to specific pathogens. So we're gonna, we predict basically these antibodies are gonna bind to the target protein. And then we add our enzyme labeled antibodies and add a substrate. And when this substrate interacts with this enzyme, it actually generates a yellow color. And what this yellow color is telling us is that there are antibodies present in the serum, which is what we predict. However, we still had to quantify this. So again, we use a plate reader software that is used by the computer, and it's gonna basically tell us the amount of antibodies we have present. And it does so using the optical density and using the absorbance. And the absorbance we use is, or the wavelengths we use was 450 nanometers to read how many antibodies we have present. And this is our data for those readings. So again, we tested this in week 4, 7, and 10. That's when we drew the blood and isolated the serum to test for the amount of antibodies that we had present. So the y-axis represents the optical density of immunoglobulin, which is the antibodies, and that was under 450 nanometers. And the y-axis represents our different groups of the mice, and this is sectioned off to week 4, 7, and 10. So as we see, this is our main focus here, which is the vaccine that we're testing. It's a vaccine that we created. And this is our negative control, and this is our positive control. So what we conclude from here is that just like our positive control, our vaccine has a high levels of optical density of the immunoglobulin, which is the antibodies that repress the pathogens coming in, and which is what we predicted. Also, what we see is the optical densities are constantly increasing, going from week 4, 7, and 10. And what that translates to is the immunity against the influenza virus is basically building stronger, so that when they're finally challenged and exposed to a live strain of influenza, they will have a stronger response against these pathogens coming into the body. So that allowed us to support our hypothesis that M2 proteins would create a suitable cross-protective virus-like particle-based influenza vaccine. And what we conclude from this whole study is that our vaccine is non-toxic in cells, and it's going to be highly immunogenic, meaning that it's going to generate an immune response in the immune system. Along with this, we conclude that the vaccine administered via the laser ablation method transdermally through the skin is going to have high levels of immunoglobulin, which is the antibody that neutralizes the pathogens coming in. And these are just some key pointers or take-home messages I wanted to make. So again, M2 proteins in influenza viruses are highly conserved, meaning they're going to stay consistent even when they're exposed to evolutionary pressures or viral genetic drifts. Along with this, M2 microparticles are non-toxic